Welcome to OLOGY. I am Nuke. Welcome to our educational video on the transcription of DNA, the blueprint of life. In this video, we will dive into the fascinating process that allows DNA to be converted into functional proteins in our cells. So, let's get started. Let's first understand what a gene is. Genes are the basic units of heredity, the building blocks that determine our traits. Eye color, height, skin complexion, all these characteristics and more are determined by our genes. Genes are segments of DNA, which is a long molecule that carries genetic information. DNA is found within the nucleus of our cells and acts like a code, containing instructions for building proteins. Gene coding occurs when the information from a gene is transcribed into messenger RNA, mRNA, which acts as a template for protein synthesis. Through the process of protein synthesis, our cells read the mRNA and construct proteins, which ultimately determine our traits and characteristics. Have you ever wondered why DNA can't simply leave the nucleus and manufacture proteins elsewhere? To understand this, let's first grasp the critical role of the nucleus. The nucleus serves as the control center of eukaryotic cells, where DNA is contained within its double membrane. It provides a protected environment for the DNA, which is vital for maintaining genetic integrity. Inside the nucleus, DNA is tightly wound around proteins called histones, forming a structure known as chromatin. This complex of DNA and proteins ensures efficient packaging and organization of the genetic material. It plays a crucial role in gene regulation and accessibility, allowing the cell to control which genes are expressed and when. DNA carries the genetic information necessary for the survival and functioning of all living organisms. However, DNA itself cannot produce proteins directly. This is where transcription comes into play. Transcription is the process by which the genetic code stored in the DNA molecule is transcribed or copied into a molecule called RNA. Specifically, messenger RNA or mRNA. This mRNA serves as a messenger that carries the genetic instructions out of the nucleus and into the cytoplasm of the cell. Once in the cytoplasm, the mRNA serves as a template for protein synthesis where amino acids are brought together to form functional proteins. Steps involved in transcription. Let's dive into the steps involved in the process of transcription. Initiation, elongation, termination, initiation. Transcription begins with the binding of an enzyme called RNA polymerase to a specific region of the DNA molecule known as the promoter. Elongation. During elongation, RNA polymerase unwinds the DNA double helix, reads the template strand, and synthesizes a complementary mRNA molecule. Termination. Transcription comes to an end when RNA polymerase reaches a termination signal on the DNA molecule, and the newly formed mRNA is released. As you all know that DNA uses the bases adenine, thymine, cytosine, and guanine, while RNA uses adenine, uracil, cytosine, and guanine in RNA adenine pairs with uracil. In prokaryotes, the transcription process occurs primarily in the cytoplasm, where the DNA directly serves as a template for RNA synthesis. The prokaryotic RNA polymerase, composed of several protein subunits, initiates transcription by binding to the promoter region of the DNA. Once bound to the promoter, the DNA is unwound, exposing a small portion of the template strand. The RNA polymerase then begins synthesizing a complementary RNA molecule, known as the primary transcript or pre-mRNA. Unlike eukaryotes, prokaryotic transcription lacks the complex processing steps seen in eukaryotic cells. Following the synthesis of the primary transcript, the prokaryotic mRNA is generally ready for translation into proteins immediately. Eukaryotes exhibit a more intricate transcription process, as their DNA is enclosed within the nucleus, separated from the cellular machinery responsible for translation. The transcription process in eukaryotes involves different types of RNA polymerases, each responsible for specific classes of genes. RNA polymerase II, the most common type in eukaryotic cells, initiates transcription by binding to the promoter region of the DNA, similar to prokaryotes. However, eukaryotic transcription requires additional protein complexes, known as transcription factors, to facilitate the proper initiation of transcription. Once transcription is initiated, RNA polymerase translocates along the DNA, unwinding it and synthesizing a complementary RNA molecule. However, unlike prokaryotes, eukaryotic primary transcripts, known as pre-mRNA, undergo several processing steps before becoming mature mRNA, ready for translation. Processing steps in eukaryotes involve the addition of a 5' cap, the removal of introns through RNA splicing, and the addition of a poly A tail at the 3' end. This intricate process ensures the removal of non-coding regions and enables the efficient translation of the genetic information. The role of RNA polymerase. Now, let's shine the spotlight on the essential enzyme responsible for transcription. RNA polymerase, RN. Polymerase is an enzyme that binds to the DNA molecule at the promoter region and catalyzes the synthesis of mRNA by adding complementary RNA nucleotides. Once mRNA is synthesized, it detaches from RNA polymerase and becomes available for protein synthesis. Points to remember: transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by enzymes called RNA polymerases, which link nucleotides to form an RNA strand, using a DNA strand as a template. Transcription has 3 stages: initiation, elongation, and termination. In eukaryotes, RNA molecules must be processed after transcription. They are spliced and have a 5' cap and poly A tail put on their ends. And there you have it. We have explored the captivating process of transcription, the reasons why DNA does not directly produce proteins, the steps involved in both prokaryotic and eukaryotic transcription, and the significance of the RNA polymerase enzyme. Thank you for joining us on this educational journey. Stay curious and keep exploring the wonders of life. If you haven't understand the concept of DNA replication, please do watch my other video on DNA replication.